And so for our second keynote to kick us off for the conference, we have Alessio from the University of Florence, who's going to tell us a more specific example.
Yeah.
So, floor is yours. You have a? Yes,
I do.
And the pointer is there. There you go.
Is there a pointer? Okay, more or less. Okay, good afternoon, everybody. So today I'm going to talk to you about the integration of reference genome and cytogenomic for biodiversity studies. So in the genomic era, we are all used to think about chromosomes as a series of nucleotides that we usually see in our nice FASTA files, right? But we all know that chromosomes are not just text file that we work with, but they are a specific structure that we have in our cells. They are made up of DNA and protein together, and they form bodies that they have their own specific morphologies, and they are present in different number in different species. And for more than 5 decades, actually, genome evolution was investigated through cytogenetics by looking at these morphologies in different species, as it was very well described in this review from Ferguson Smith, History and Evolution of Cytogenetics. And today also cytogenetics offers scientists a valuable set of tools to investigate the evolution of chromosomes. For instance, you have this nice review in chromosome evolution in marsupials that you see here that was recently published, and then you have the other example on the right about the study of sex evolution and sex chromosomal rearrangement in Sauropsida and mammals that was a pillar for the understanding of evolution of this set of chromosomes in this group of vertebrates. So these 5 decades of data actually has something that we should not toss away, that we should not forget about, because they're offering us a valuable set of information to connect to our genomic data nowadays, okay? But there is this small problem of miscommunication that we have today about the connection between genomic data that you have on the left and the cytogenetic data that you have on the right. I mean, I will explain this with an example. This is Emys orbicularis. This is a European pond turtle species. We actually assemble the genome of this species and we produce the karyotype of this species, which is that one that you see there. As you can see, the karyotype of many turtles, and for sure of all emidids, in those karyotype chromosomes are not listed by size, are not ordered by size. They are ordered by morphology. You first have the metacentric chromosome, then you have the acrocentric chromosome, and then you have the microchromosomes, okay? This is conventional for turtles, and all the cytogenetic data you have on turtles are based on this, okay? And this is what you see on the genome assembly. As you can see, this is an assembly that we produce chromosome level. It perfectly matches the 25 pairs of chromosomes of the species, but you have this small problem that chromosomes here are listed by size. So what is chromosome 7 in our karyotype does not necessarily, I mean, it does not match the chromosome 7 of the genome assembly, okay? So basically this gap make you miss all the data you have gained and until 2025, which is the year we published the genome, based on molecular cytogenetic data, okay? So actually, how can we link these two bunch of data? How can we link cytogenetic and genome? How can we get to the cytogenomic level? Well, actually, you know, we have, sorry, I forgot to mention this review where Lewin, Graves, Steve O'Brien, and all other big names on cytogenomics actually explain very well this problem. So, as I was said, we have some technologies that link the chromosome structure to the genome. We have IC technologies that you know of course, but this doesn't really create a link between the genome and the karyotype. This is just something that tells us how nicely a bunch of assembled genomes interact each other. So this is something that tell us how to better scaffold our genome, but does not create any link. Well, actually, there is a technique which is single chromosome sequencing that allow us to connect a link between the karyotype and the genome. So single chromosome sequencing is very simple to understand. You basically physically isolate your chromosome, and then you sequence the genetic material of your chromosome, okay? So the sequencing, it's easy. It's just a normal sequencing in short reads usually. The difficult part is the physical isolation of the chromosome. You can do this either via cytofluorimetry with a flow sorting or with a microdissection if you have enough patients to do that, okay? I will spend just 5 minutes on the description of these two techniques because I think they're not very common and because I think they're cool. So I will spend a couple of words. This is a microdissection apparatus. This is an inverted microscope. So you have objective down there, and then you have a micromanipulator installed on the microscope with two arms. You have a needles, which is used to scratch your chromosome, and you have a pipette in which you collect your chromosome, just like this. So you have your metaphase there, and you have your microneedle. You scratch the chromosome, and then you put the chromosome inside the pipette. And once the chromosome is inside the pipette, you can sequence everything, and then you have your reads of your chromosome. Okay, so this is should be a video that show you how it happens. So you literally scratch the chromosome out of your metaphase. This is a metaphase of a frog. Then you pick your chromosome. They are quite sticky, and then you insert the needle in your pipette, and then in that you broke the pipette into the tube, and then you sequence the material, okay? You have another method, physical method to isolate chromosome, which is laser microdissection, which is basically works the same with a laser. You just cut the microchromes out of your metaphase, and then you put it into a small tube. As you can see in this other video, you can physically isolate the chromosome with a software. Then the laser cut the chromosome, and it shut the chromosome inside the tube. And in this case, you can barely see the difference, but okay. The other technique for chromosome isolation is the chromosome sorting. This is quite different stuff because it requires a huge amount of cell cultures. So you have to start from a proper amount of tissue source. You don't work with slides. In this case, you work with chromosome suspension, okay? The chromosome suspension is then run into a flow cytometer. The flow cytometer can isolate the chromosomes based on their charge, which is basically based on their size, okay? And then the isolated chromosomes are then used to perform a fluorescence in situ hybridization on a metaphase. In this case, the chromosome can be recognized as the chromosome of your karyotype, and then in the end, you have your isolated chromosomes with their names, okay? This is more laborious than the microdissection, but more effective as you can understand from this picture. You can gain more copies of the same chromosome and then more material to sequence, okay? So apart from all of this stuff, the idea is that when you have your isolated chromosome, you perform a whole genome amplification to amplify your genetic material. Then you sequence everything, and then you map everything onto your genome. We usually do this with the DOPSEC pipelines, which are some pipelines developed by Vladimir and Alex at the Russian Academy of Science in Novosibirsk. Now, nor Vladimir and Alex, they're not there anymore. But anyway, the pipeline was developed by them. And so the idea is basically this one. You have your reads, usually short reads of your chromosomes, of your real chromosomes, because you isolated them, and then you have the reads of your chromosomes. Then you have the scaffold of your reference genome, or whatever, how you want to call them. Then you map the chromosome reads onto the scaffold of your reference genome, and then you fish out the information of the reference genome. So by a simple mapping with some refinement, you can find the scaffolds that physically correspond to the chromosome of your karyotype, okay? So you can find this link, this connection. This is all very beautiful, but how can we use ChromSec data for the study of biodiversity conservation? So ChromSec was widely used to assist genome assembly, and I will show you some example in a minute about this. Chromosome sequencing and cytogenomics in general was used to assist genome evolution studies. We have a lot of examples in which cytogenomics was particularly useful for some cases about the study of genome evolution in many vertebrate species. And cytogenomics can actually be used also for biodiversity conservation, and I will show you some examples of this as well. So starting from the first one, for those of you who are working with reference genome, vertebrate reference genome from the very beginning, you may recall this paper. This was one of the first reference genome that came out in 2012. For sure, was one of the first of marsupial reference genomes. And in this case, chromosome sequencing was used to assign 99% of the genome to the karyotype of the species. And as I will show you in a couple of slides, this was particularly useful to identify the genomic region connected with the facial tumor disease of the species. But also, from an evolutionary point of view, the important thing here was that by means of this technique, they managed to produce the first comparative map between the devil genome and the opossum karyotype and the opossum genome, which was the only other marsupial for which genomic data were available. Again, different group of vertebrates, reptile, the green anole lizard was the first genome of reptiles that came out. And also in this case, in 2016, with a different paper, some important refinements were made on the genome to assign the genome to the karyotype. And in this case, these lead authors to understanding some very important, to do some important consideration on the evolution of sex chromosome in anole lizard. And they identified some neo X and Y chromosomes, which could not be identified only on a genomic basis. Now, as you can see, these two papers that I mentioned, the green anole and the Tasmanian devil, were quite old. Nowadays, today, chromosome sequencing is still used to assist genome assemblies of those complicated species. For instance, Ascipenseridae, sturgeons are particularly complicated from a ploidy point of view. The karyotype there is the karyotype of Siberian sturgeon, and as you can see, it is octaploid. So as you can see, the metaphase there, it's quite difficult to assemble the genome of such species. And I show you this example here of the sterlet sturgeon genome, which was published in 2020 on Nature Ecology and Evolution. And here a microdissection approach was used to identify homology between within the polyploid genome of this species. Somebody very with a lot of patience microdissected all the chromosome of the metaphase. Other difficult species are all those species that has a large genome. For instance, the Ambystoma mexicanum has a genome of 32 gigabases. And in this case, the initial characterization of the genome was actually done using a microdissection approach. In this case, it was laser microdissection. And authors managed to do some partition of the genome and assemble the genome and try to produce a draft based on data they gained with this technique. Another nice example is this one. This is a paper we contributed to, the pangenome of the barn swallow. In this case, the cytogenetic approach was used to guide IC data. So the IC data is quite difficult to interpret when you have microchromosomes, so in birds, of course, and some reptiles also. So in this case, in the pangenome, we missed a couple of chromosomes that actually were present in the karyotype. So based on the karyotype data, we managed to rearrange, to better curate to do a better manual curation of the pangenome. Last example I'm showing you about the ChromSec to assist the genome assembly. This is the example I showed you before in the beginning. So we performed some preliminary chromosome sequencing analysis of the species that they are already published in that paper that came out a couple of months ago. And as you can see, we managed to microdissect 6 of the macrochromosome of the species. And as you can see here, as I told you before, chromosome 7 do not correspond at all to chromosome 7 of our genome assembly, but actually is chromosome 12 of our assembly. And this made us do some consideration about the evolution of the acrocentric and metacentric chromosome in immediate turtles based on previous cytogenetic data of the group. As I said, a cytogenomic approach can be used to assist genome evolution studies. I don't have the time now to show you example about the evolutionary studies in which cytogenomics gave a big contribution. I just want to mention these nice reviews here, in which you can see cytogenomics helped the study of genome evolution from fish to reptiles, marsupials, human. Chromosomics is just another term to define cytogenomics. So this science that tried to create the link between the genomic data and the cytogenetic data. But is all this useful also for biodiversity conservation? Well, actually, there are some authors like Potter and Deakin that stated in this paper that cytogenetics should be included in the conservation genetics toolbox. And I think it's something very nice to mention about this technique, is that it can improve species identification and taxonomy. It can help the detection of chromosomal abnormalities associated with inbreeding and disease. And I will show you, and here we will go back to the Tasmanian devil. It can help the study of sex chromosome and sex determination system. And we will see that this is extremely important for species that relies on ex situ conservation program. And of course, cytogenetic and cytogenomic can help also the definition of management units in the so-called population cytogenetics. Starting from this point here, we have this nice example, authors cite this nice example in their review about the wallabies. This species is distributed on the east coast of Australia, and no genomic data were available. I mean, only a little about some markers. And with an integration of cytogenetic data and those little genetic information, they actually managed to define a gradient of, well, in some case species, in some other case subspecies, and in some other case management units along the cost. So in this case, cytogenetic was particularly useful for the determination of management units in the species. Same thing was for these fish in two islands in front of Brazil. In this case, no genomic information were available, but authors managed to integrate cytogenetic data, and they actually found two population of the same species with huge chromosomal rearrangement that made them define two different management units. Again, this is the infamous tumor facial disease in the Tasmanian devil, and by the use of a cytogenomic approach, authors here managed to identify the genomic region involved in this disease, and also that they were linked to sex chromosomes. So again, cytogenomic here was pretty useful for the conservation of the species in this sense. As I said, another important application of cytogenetic and cytogenomics for conservation program are about this, about the understanding of sex determination system. Again, we are with sturgeons again, as 80% of sturgeon conservation program is made ex situ, okay? So it's not made in situ, so they rely for instance, our Adriatic sturgeon, our Italian sturgeon, is now living basically only in captivity, and they rely on the understanding of the sex determination and sex chromosome system to produce a more, let's say, to aid the reproduction of the species. And sturgeon is just an example, but in fish and in polyploid species in general, this is of extreme importance. And not only in fish, in 2022, the microdissection of the Y chromosome of the panda, which is another species that relies a lot on ex situ conservation program, allowed authors to identify some fertility-related genes that were of extreme importance for the design of conservation program for the species. Last but not least, our Komodo dragon, the species that Claudio is studying for more than 20 years, and I'm taking on with the genomic part. The Komodo dragon, I will just spend last 5 minutes, 2 minutes on this point here. So Komodo dragon lives only in Indonesia, in the Lesser Sunda Island. In 2019, we isolated the chromosome of the Komodo dragon using flow cytometry, and we produced the first reference genome for the species using a mix of NGS technologies. We produced a nice comparative map of the Varanus comodoensis with the chicken and the Anolis carolinensis, and we made a lot of evolutionary evolutionary discovery about the sex chromosome system of the Komodo dragon. Especially, we managed to finally identify the region of the genome which is linked to the Z chromosome. And this was quite useful in our preliminary genomic survey of the species in its biogeographical distribution, because we sequenced 24 individuals with a whole genome sequencing approach. And as you know, many reptiles, and Komodo dragon has a ZW chromosome system. And when we came to calculate the coverage for the Z chromosome, of course, we identify half coverage for the females and double the coverage for the males. And this is very important for the species, as the sex marker that were present in the literature were basically non-working or working only with certain kind of samples. But in this case, we managed to identify a whole region that can be used to identify the sex of the species. Last two slides. we are now working on a SNP panel that consider only autosomal chromosomes. So we managed to exclude the sex chromosome, and based on this SNP panel, which are 96 SNPs in total, we managed to identify we managed to distinguish all the 6 population of the Komodo dragon very well, and this will be extremely useful, and it's already now to assign the population of confiscated individuals that are present, unfortunately, around. So with this, but this is another story actually. So I conclude by thanking you all for your, with the take-home message. So, which is very simple, that in the genomic era, chromosomes still exist, and sometimes it's worth to have a look at them. And I thank you all for your attention, and the organizer for having me here.
Thank you very much, Alessio, for that reminder, as we are in the genomics era, that sometimes we have to rely on some of our former technologies to see the unseen. Questions for Alessio? You and then you. Yeah. Go ahead.
Can I go first?
Yeah, go first.
Thanks for the, can you hear me? Yeah, thanks for the really interesting talk. As Rob mentioned, I think sometimes cytogenetics is a bit old school, but you showed really nicely how interesting it is in combination with genomics. How big is the cytogenetics community? Because I feel like it's actually techniques you need to, know how to do this in the lab. And I feel like this is a bit like taxonomy sometimes, something that is not so exciting for students. I mean, can you expand a bit on that?
Yeah, sure. Yeah, well, actually, it's a club. Club. You have the cytogenetic club and the more open cytogenomic club, let's say. No, I'm kidding. But it's something that requires a lot of, let's say, skills that are not always written in books, but they are made of protocols that you find from very old professor, then they give you the protocol. And this, but this is true because those protocols react completely different from species to species. So you have to have your own expertise. So, but actually, there is a very active community of cytogeneticists in Europe, in East Europe. They do a lot of this work, where they actually put a lot of effort also in training students. But when I was a PhD student, I did a lot of this in the lab, and I found it quite exciting because you can see a lot of things going on. You can see what you're really working with, and I find it even more exciting when I managed to connect them with the genomic data. So you can see something on the microscope, and then you can translate them into your assembly. And this is, apart from being exciting, I think it's very useful because I found, for example, I mean, I work a lot with reptiles, and for reptiles there is a very old school of cytogenetic, also in Italy actually, and in Prague, in Czech Republic. And I found very useful some papers from the 90s that were just forgot in the literature, and I found it useful to understand some, let's say, connection that I found between birds and reptile genomes that we couldn't explain just looking at sequences. So I think it has room for development.
Thanks. Another question there?
Any information about the application of this approach to plants?
Yes. Okay. No. Plants is a completely different story. I actually had the opportunity to visit some very nice labs which were completely devoted to plants, and they didn't know anything about animals as protocols. And there is this big gap between cytogenetic, animal cytogenetic and plant cytogenetic, because protocols are completely different. With animals, you work with cells, you work with tissues. With plants, you work with I forgot the word in English, with the roots, with the top of the roots. So it's completely different protocol, but in the end, actually, plant cytogenomic was the first to came out instead of the animal cytogenomics. So there was a pioneer group also in Czech Republic where they did a lot of combination also with bio-nanotechnology. They first combined the cytogenomic and bio-nanotechnology to scaffold the genome. So actually, the cytogenomic, it's a more common point between the two kind of world, animal and plant.
Okay, I think to stick on time, we should move on. So thank you very much, Alessio.